Hello dear students in grade 8, this time for biology. First, I will solve with you your homework, then we'll complete our lesson. Now go to the page 118 here. The question said, label the rest of the marker molecules and clumping proteins in the table. As you can see here, the red blood cells contain marker A, and in his plasma contain anti-B, clumping protein anti-B. Now here of course this marker for type B, here you have to write B. Here in his plasma or her plasma contain anti-A, antibody A, right here, antibody A. Now here, blood type AB contain both type of markers, type A marker A, here marker B, here A, here B, here A, here B. Here, of course, in his or her plasma, no clumping proteins, Type O, here, no marker on the surface of red blood cells, RBC. So, in his or her plasma, that means contain both clumping A protein and clumping B protein. Contain antibody A and antibody B. Now go to the next page. Here, summary in the graphic organizer here. Complete the main idea about safe transfusion. There write the supporting details here. Marker molecules on your red blood cells. Determine, of course, the marker in your blood cells on the surface of your blood cells. Determine your blood type and type of blood you can get in a transfusion. So here, the marker molecules determine your blood type and type of blood you can get or receive from another donor. Details here. Type A, for example, can receive type A or O. Type A, for example, and O don't have B marker. Type AB can receive all types. I repeat, here type A can receive type A or type O. Here type A and O don't have B marker. Here type AB can receive all types. Now here question number 2, which blood type does a person have if the person can receive only the same blood type for a safe transfusion? Explain. Of course here type O only receive from type O. Why? Because it will recognize markers in type A and B, because the person who has type O has clumping protein or antibodies A and B. Now we'll start to revise our lesson, then we'll complete it. As you can see here, our blood has 4 main components: plasma, red blood cells, white blood cells, and platelets. About 45% of your volume of blood is cells. 55% is a liquid, plasma. Now, as you can see here the white blood cells, here platelets, here red blood cells, and here white blood cells. So what is a plasma? Plasma is a yellow liquid forming about 55% from the volume of all the blood. Most materials carried by plasma, in addition, carrying like nutrients, glucose, vitamins, hormones or signal chemical signals. Also, plasma carry away from cells to the other system carbon dioxide uh, and wastes. Of course, uh, white blood cells and so on. White blood cells are produced in bone marrow. We'll talk later. So what is the function of red blood cells? Here, red blood cells carry oxygen from the lungs to cells throughout the body. Of course, the red blood cells made up from protein called hemoglobin that contain iron that help to bind oxygen to it. Red blood cells produced in red bone marrow. Now, white blood cells consider the first line for defending, or we use white blood cells to defend about the body from any bacteria or any viruses. So white blood cells fight disease by alerting. This is the first type of the white blood cells, alerting body to invading organism, or that this is the second type of white blood cell producing chemicals to fight the invader, and this is the third type of white blood cell surrounding and killing the organisms like bacteria and viruses. Here, white blood cells combined with red blood cells bigger a little bit. We can found only one white blood cells between from 500 to 1000 red blood cells. Finally, platelets helping to form clots or help to form blood clots by producing protein. We called it fibrin. We called it fibrin that waves on it and block the wound. Also, we studied that the scientist is named Carl Landsteiner studied what the blood and discovered the four types of the blood. I said to you, Carl Steiner discovered three proteins now that determine the type of the blood. The first one, we called it marker. This protein found on the surface of the red blood cells. This marker or this protein determines the blood type and determine the type of the blood that you can receive from donor. So marker molecules on your red blood cells determine your blood type and type of blood that you can safely receive from another person in transfusion. Here, this person receive blood. As we can see here, we have four types of the blood: type A, type B, type AB, and type O. Type O. Type O. Here also we can see the marker here. Type A determine the red blood cells. Here B determine the red blood cells for B. Here this red blood contain both type A and B. O doesn't contain any marker. Now let's see video now here to summarize the blood components and types of the blood.
Red blood cells are constantly moving through your body, delivering oxygen to your cells.
As we can see here, the red blood cells back to the lungs to exchange gases. Then here blood cells carry oxygen and move to the oxygen here.
Red blood cells, along with the other components that make up blood, move through blood vessels in your body.
Here exchange what material with the cells or tissues of the body.
Red blood cells travel in your bloodstream along with white blood cells. White blood cells are the body's disease fighters.
Yes, here white blood cells, here you can see red blood cells, and there you can see platelets. So we can see red blood cells moving in bloodstream.
Red blood cells also travel with platelets. Platelets are cell fragments that help form blood clots. When one of your blood vessels is damaged, some red blood cells will become part of the clot that forms.
Note here, the plates come and start to stick on the sides of the wound and form fibrin that waves the net.
Red blood cells, along with the other components of blood, float freely in plasma. Plasma is the liquid part of blood.
Here we can see the function of the plasma. As I said it before this time, sorry. Now,
Red blood cells are constantly moving through your body, delivering oxygen to your cells. Red blood cells are constantly moving through your body, delivering oxygen to your cells. Red blood cells are constantly moving through your body, delivering oxygen to your cells.
now as I said to you, we have four types of the blood. Blood A, that carry marker A. Blood B, that carry marker B. Blood AB carry both marker A and B. Marker, remember, determine the blood type and determine the type of the blood that can you safely receive. Now, clumping protein. What are the clumping proteins? I said clumping proteins found in the plasma. Clumping proteins recognize red blood cells. No red blood cells. So any foreign or any blood cell coming from another person, here clumping protein, you will know it. Only the clumping proteins know the red blood cells. Any foreign red blood cells come from anyone, the clumping protein will develop and interact with these new red blood cells. So clumping proteins in your plasma recognize red blood cells with foreign markers that are not yours. As we studied before that, here type A contain antibody, or we call it clumping protein B. That means if any blood type from B found here, the clumping protein here will react with B and causing to type B to clot. Here, type B contain antibody or clumping protein A that react with what? Interact or react or causing to the type A to what? To clot. AB doesn't have any clumping proteins or contain both A and B proteins, so cannot this type or cannot receive only from O. AB can receive from any type because no clumping protein. As we can see, the type O can give A and can give AB and can give B, also can give itself. A can give itself only, can give only AB. B can give only the same type, also can give AB, because here no clumping protein. AB can take from AB, can take from B, can take from O, can take from A. So type O can give all the types, but type AB can take from any type. Now we'll complete and we'll study the third factor that the Steiner determine and help us to save transfusion. Of course, the Steiner studied another protein found on the surface of red blood cells. We call it rhesus or RH, rhesus factor or RH factor. Of course, this protein limit compatibility to receive blood from anyone. For example, Steiner studied many people. He found about 85% where has this protein. So he said has rhesus or RH positive blood or RH positive. That means their red blood cells contain this protein or this factor. A person without it has negative blood. A few person or a few people don't have this factor, so we call it RH negative blood or RH minus. Of course, the process called cross matching is used to ensure blood of compatible type is used. For example, person who has this protein factor RH cannot receive any blood from this person that doesn't contain this protein, because this protein or RH make the clumping protein develop, react quickly, as I said to you. So only here the person who has type blood or has factors can receive from the same person who has positive or has the same protein here. Now watch this video to understand.
A blood type refers to the presence or absence of a certain marker or antigen on the surface of a person's red blood cells. For example, in the ABO system, presence of A or B antigen gives type A or B, presence of both antigens gives type AB, while their absence gives type O. Blood typing is critical for blood transfusion, as there are very specific ways in which blood types must be matched between the donor and recipient for a safe transfusion. The rule is simple. Patients should not be given antigens that their own blood does not have. This is because the recipient's immune system may recognize any new antigen as foreign and develop antibodies to target it for destruction. Depending on the scale of the triggered immune response, the reaction can be serious or fatal. Applying the rule, a type A patient who is negative for B antigen can only receive blood from type A and type O donors whose blood does not contain B antigen. A type AB patient having both antigens can receive blood from anyone, while a type O person being negative for both A and B can only receive from type O donors, but can give blood to anyone. Another important system is the Rh system, for which D antigen or Rh factor is best known. The blood type for this antigen can be either Rh positive or Rh negative. By the same rule, a Rh negative patient cannot receive blood from a Rh positive donor, while the reverse direction is fine. Each of the 4 types of the ABO system can be Rh positive or negative. This gives 8 possible combinations, the 8 basic blood types everyone knows about. But ABO and Rh are only a fraction of the 35 currently known blood group systems, many of which can cause serious reactions during transfusion if mismatched. Altogether, there are hundreds of antigens, giving rise to a gigantic number of possible blood types. A fully specified blood type should describe the complete set of antigens that a person has. In theory, this list must be determined for both donor and recipient before a transfusion can take place. In reality, however, most people only need to care about their ABO type and Rh factor. The ABO and Rh systems are the most important in blood transfusion for 2 reasons. First, most people can produce robust antibodies against A, B, and D antigens, which
So, to transfer the blood for donor to recipient, you have to know the type of the blood, the Rh factor. So the marker that we studied that determine the blood types and the clumping proteins that help us to determine which blood type can receive, and Rh factor also help us to compatible between the donor and recipient persons.
may not be the case for other antigens. In fact, anti-A and anti-B antibodies are usually developed during the first year of life. Second, the 8 basic blood types are distributed in comparable proportions that makes mismatching a likely event. Most other antigens occur at such frequencies that only a very small subset
Here we have a table for safe transfusion, red blood cells compatibility table. Here we have donor and we have recipients. For example, the donor here who has type O but doesn't have Rh factor can give this one and can give all the other types. Now, here, the other person has blood cells O, type blood cells O, and carry Rh factors, cannot give what another type O, but that cannot. Why? Because this Rh what develop the clumping protein. Develop the clumping protein in the plasma of recipient and causing to the type O plus to clot. Can give here O and can give plus. As you can see, can give only the person who has Rh factor. Here, as you can see, the donor that have factor here type blood A and has Rh factor can give only the A that has and can give AB that only has Rh factor. Now, finally, we'll start to study the lymphatic system. The lymphatic system. Finally, we study the lymphatic system. We studied that the red blood cells or the blood carry nutrients, minerals, water, and many things to the cells of your body. Now, here during the exchange in capillaries between the cells and bloodstream, sometimes extra fluid will move through the spaces between tissues or cells. After the fluid move into the cell, some fluid or extra fluid will stay between the cells and tissues. Here we have another system return this fluid into bloodstream. This system we call it drainage system, we call it lymphatic system. As you can see here we have lymphatic system. So lymphatic system is a drainage system like forming a network. It has what? Vein-like vessels. Vein-like vessels. What is the function of lymphatic system? Return or absorb the fluid, extra fluid that stay within the tissues into bloodstream. After materials are absorbed in tissue through diffusion, excess fluid returns to bloodstream through the vessels. Of course, the fluid contain water, contain white blood cells, contain glucose, contain many things. Now, the fluid that move within the lymphatic system, we called it lymph. So here the fluid inside lymphatic system is called lymph. Lymph consists of from water, dissolved material, glucose, and white blood cells. Here, as we can see here we have like knobs, or this area we call it lymphatic nodes. Lymphatic nodes here help to filter out what? To filter out the bacteria and viruses from lymph or fluid before it returns to the bloodstream. That means the lymphatic system interact between circulatory system and between immuno system. That means help to return the fluid into the bloodstream and help to what? To fighting disease, especially in lymph nodes. So lymph nodes is a small knobs of tissue which filter lymph from trapping bacteria and other disease-causing microorganisms in the fluid. Finally, lymphatic system help to return the excess fluid from tissues and cells and filter it out from bacteria, then return back to the bloodstream. Now see this video about the lymphatic system.
In a nutshell, the lymphatic system is a drainage system that removes excess fluid from body tissues and returns it to the bloodstream. It's actually a subsystem of both the circulatory and immune system. The major purpose of the circulatory system is to bring oxygen and nutrients to body tissues and remove wastes. This exchange happens in the smallest blood vessels called the capillaries. Blood plasma containing nutrients moves out of capillaries at the arterial end of capillary beds, while tissue fluid containing wastes reabsorbs back in at the venous end. However, not all of the fluid is drawn back to the bloodstream at this point. About 15% of it is left in the tissues and would cause swelling if accumulated. This is where the lymphatic system comes into play. It picks up the excess fluid
and returns it to the circulatory system. Unlike the blood circulatory system, which is a closed loop, the lymphatic system is a one-direction, open-ended network of vessels. Lymphatic vessels begin as lymphatic capillaries made of overlapping endothelial cells. The overlapping flaps function as a one-way valve.
So the fluid that drains to the lymphatic system found in the area between cells and tissues here.
When fluid accumulates in the tissue, interstitial pressure increases, pushing the flaps inward, opening the gaps between cells, allowing fluid to flow in. As pressure inside the capillary increases, the endothelial cells are pressed outward, closing the gaps, thus preventing backflow. Unlike blood capillaries, the gaps in lymphatic capillaries are so large that they allow bacteria, immune cells such as macrophages, and other large particles to enter. This makes the lymphatic system a useful way for large particles to reach the bloodstream. It is used, for example,
Here the gap between the capillaries here allows the bacteria to move inside the lymphatic system. When the lymphatic drain was the fluid again to the bloodstream.
for dietary fat absorption in the intestine. Once inside lymphatic vessels, the recovered fluid is called lymph. Lymph flow is enabled by the same forces that facilitate blood flow in the veins. It goes from lymphatic capillaries to larger and larger lymphatic vessels and eventually drains into the bloodstream via the subclavian veins. On the way, it passes through a number of lymph nodes, which serve as filters, cleansing the fluid before it reaches the bloodstream. Lymph nodes are small bean-shaped structures scattered throughout the lymphatic network. They are most prominent in the areas where the vessels converge. Lymph nodes contain macrophages and dendritic cells that directly swallow up any pathogens, such as bacteria or viruses that may have been taken up from an infected tissue.
Now, finally, I need from you to solve your homework, page 120, page 121, page 122, page 123. See you later, inshallah. We'll start with next week, inshallah. We'll start with the new lesson.